Topoisomerases. Before talking about topoisomerases, let's discuss about the supercoiling. The supercoiling occurs when the molecules relieves the helical stress by twisting around itself. The over-twisting leads to positive supercoiling and under-twisting leads to negative supercoiling. The length of the DNA can be 1000 times to that of the cell. So that the supercoiling is very important to pack the DNA intactly in a small volume of the cell. The entangled strands, when get separated, increases the torsional stress, creating more tangles. This takes place in the DNA always. So these problems need to be tackled. It is done with the help of DNA topoisomerases. So what are DNA topoisomerases? The topoisomerases are the class of enzyme that alters the supercoiling of double-stranded DNA. That is, they act by cutting one or both strands of DNA. This break allows the DNA to untangle or unbound. There are two classes of topoisomerase, that is topoisomerase type 1 and 2. First, we will discuss about topoisomerase type 1. The type 1 topoisomerases is also known as nicking closing enzymes. They are monomeric unit with 100 kDa molecular weight. They are present in both prokaryotes and eukaryotes. They remove the negative supercoilings without leaving any nicks in the DNA molecule. And they does not require ATP hydrolysis. Let's discuss about the mechanism of topoisomerase 1. The mechanism involves the formation of covalent bond between tyrosine residue and the phosphodiester backbone. The nucleophilic attack from the hydroxyl group of the tyrosine to the phosphorus atom of the DNA creates a phosphodiester link between the enzyme and the DNA. This will generate a free 5'OH group. The other strand is held in place by binding non-covalently to a domain of the enzyme. The strand is thus resealed. The topoisomerase 1 of E. coli will act only on the negative supercoils, whereas topoisomerase 1 of eukaryotes will act on both positive and negative supercoilings. Now comes the topoisomerase 2. The topoisomerase 2 are 375 kDa protein that consists of two pairs of subunits that is designated as A and B. The enzyme which cut on both strands of DNA helix simultaneously in order to manage tangles and supercoils. They unknot and untangles DNA by passing an intact helix through a transient double-stranded break that it generates in a separate helix. It requires ATP hydrolysis. The mechanism of topoisomerase 2. It can cut both strands of DNA and pass another strand of DNA through it and can join by utilizing the ATP. The double-stranded DNA is further twisted. This twisted region of the supercoils is called as the right region. The topoisomerase 2 will unbound these positive supercoilings by introducing negative supercoilings to it. By introducing negative supercoilings, it is done by gyrase, which will introduce the negative supercoilings by 2 units. It will cleave the both strand of DNA and will pass the whole other double-stranded DNA through it and relegate it. Thank you.